Hello dear students, I am back with the same chapter, that is reproduction in flowering plant, okay, for the class 12 biology. Okay, we will have a quick revision of the things that we have seen in previous video or previous lecture. Okay, previously we have started with the post fertilization event. Okay, in post fertilization event we have started with embryo as well as endosperm we have completed. Okay, before that we have seen with pre-fertilization step. Okay, in pre-fertilization we have seen with male gametophyte and female gametophyte structure we have seen with. Okay, then on we have also seen with pollination part and agents used for pollination in pre-fertilization. Okay, and then we have seen with fertilization, wherein we have seen with the double fertilization, how a double fertilization process which is only unique in plant was seen. Now in post fertilization event we have seen with endosperm. Okay, so endosperm formation is what? It is a result of primary endosperm nucleus formation. Okay, this were again dividing to primary endosperm cell and then primary endosperm tissue was seen. But it is said that endosperm is what? It is a nutritive cell which provides nutrition as well as protection to embryo. Okay, so protection has to be given. Suppose this is an embryo, but to this a protection is given. This protection is nothing but it is given by the endosperm. So before embryo formation, endosperm formation is evolved. इसका ज़रूरत हो गया कि पहले endosperm form होगा तो ही वो provide करेगा nutrition to embryo. Now endosperm formation based on mode of formation, it is divided into nuclear, cellular and halobial. Okay, we will see with the new term. This is halobial mode of nutrition, mode of formation of endosperm. Halobial is an intermediate for the nuclear as well as cellular. ये दोनों का जो मिश्रण होता है that is halobial. Okay, now nuclear, इसके लिए first we will see with the example first. Okay, suppose this is a nucleus. Okay, the polar nuclei, the two nuclei. Okay, I hope you remember with the diagram. I have told you the three antipodal cell over here at the chalazial and synergid cells, egg cell and two polar nuclei. This were forming what? Endosperm. So ये जो nucleus है at one, this is nucleus or nuclear division. This divides, which will be the cellular one. Okay, further they mitotically divide to form a cell wall at the periphery. Periphery in the sense corner पे, border पे. Okay, this cell formation, that is mitotically they are dividing. Okay, once the mitotic division occurs, the third, which is called as halobial or free nuclei formation. Here in third step we will see, that is this one. Okay, these cells are there and in it you see the free nuclei. ये है nuclei. Okay, I hope you are clear with the three stages or three mode of formation which leads to endosperm. See, first nucleus is dividing and then cell wall formation at the periphery of a cell, then the free nuclei formation. The best example for this I have told you yesterday, that is coconut. जो brown color part you see of coconut, this is nucleus, brown color part, and when the whitish matter that you see, the white color material, that is this one, cellular division. Okay, and then the water, जो पानी होता है, that is this free nuclei. There are thousands of free nuclei formation. Okay, I hope you are clear with the endosperm. Easiest we have told, nuclear division or nuclear endosperm, cellular endosperm and halobial endosperm. Okay, for coconut brown color part, white color का जो अंदर का होता है coconut वो हो गया cellular endosperm, and then the water, that is free nuclei. Okay, next we will see with the I think we have also started with the embryo. The second in post fertilization is embryo. Embryo formation is a result from zygote. Okay. Once the zygote are formed, from zygote it develops to embryo. Zygote formation is a result of one male gamete fusing with the egg cell, that is leading to zygote formation. Now zygote when fertilized, it is having its difference in dicot as well as in monocot. Okay, the embryo sac formation or embryology or embryogeny. Okay, it is a process of development of embryo, embryogeny, okay, or embryology. Then other term also I am writing, embryogeny. Now we will see with how embryo are developed in dicot. See with the diagram carefully I am drawing with of dicot mostly. Suppose this is a zygote. Okay, this zygote divides in both axes. Okay, it divides in nuclear axis also to form two different or two irregular shaped cells. The top one is called as suspensor cell and this is embryo sac or embryo cell. Okay. Now suspensor is also called as stalk, which divides transversely, whereas embryo further divides transversely as well as vertically. Okay. So this is the suspensor cell which divides to 6 to 8 cells or sometimes 6 to 10 cells. Okay, 6 to 10 cells and it divides, and this divides in 2 tier. Okay, at last I will tell you what is 2 tier 8 cell. Okay, now this divides like this. From the 2 it is forming 4 cell. Okay, the first stage, this is called as proembryo. Okay, proembryo this is, and then the next, this is suspensor. Again this suspensor cell will divide to 6 to 8 or 6 to 10 cells in it. Please see with the diagram and then you can match out with the diagram which is given in your textbook. Okay, this again grows like this, 1 2 3 and 4, 2 3, 1 2 3 4 5 6. See like this, I will draw again. This is called as 2 tier 8 cell. You can see over here 8 cells are there which are in 2 different parts. This is called as 1 tier 4 cell. Okay, this type of development is called as globular. Okay, next is in the same division we have seen that it divides in somewhat heart shape. The suspensor cell also divides and it forms some heart shaped cell, heart shaped, which is having again cells growing in it at the periphery. This is how an embryo develops. Okay, and then at last the third one will be, I am drawing it over this side. Okay, this is somewhat mature form. Mature form of embryo I am drawing it. Okay, the suspensor somewhat like this it looks. This line you see, that is called as cotyledon, and this forms radicle and this forms okay, mule. This is how a mature embryo looks in dicot. I hope you are clear with the diagram and the stages of development of embryo. First, it is dividing and forming two different cells, that is suspensor and embryo, and then it is forming pro-embryo stage, globular, pro-embryo, globular, heart shape, and then at last it forms mature embryo. Okay. This development is seen in dicot. Same thing we have in monocot also, but it does not have globular and heart shape. Okay. This was all regarding embryo. Embryo get nourishment from endosperm. Okay. Now we will see with the next part, that is formation of seed. Okay. In formation of seed also, in monocot it is different and in dicot it is different. Okay. Before seeing with the formation of seed, seed are evolved from, they are evolved from, or they are formed from ovules, and fruits are evolved from ovary. Okay. It is evolved from ovary. Now we will see, before saying this, we will see with before fertilization, this is important one, before fertilization and after fertilization event. After fertilization event, this will help you to know how the seeds are formed. Okay. Before fertilization and after fertilization, in this we will see with the first one. I hope you remember with calyx, corolla, stamen, stigma, okay, stigma, style and androecium part. Okay. They further withers, wither in the sense they fall off. They fall off after fertilization. Before fertilization they are present in a flower, but after fertilization they fall off, that is withering. But it is seen that in Solanaceae family, calyx persists for some times. Okay. Second one, for funicle, we will see with, it also remains. Third, we will see with integuments. I hope you remember with the integuments, when we have seen with the structure of ovules, somewhat like this it was having. If you remember with this, the structure I have drawn you, chalazial and micropilar, and this were called as integuments. Okay. Integuments provides seed coat, and it is seen that in some it is edible part also. Okay. In some it is edible part. इसका एग्जाम्पल एडिबल पार्ट जो खा सकते हैं, पोमग्रेनेट, जो अनार बोलते हैं, जो दाने होते हैं अनार के, दे आर नथिंग बट द इंटीग्यूमेंट्स. ओके. देन इंटीग्यूमेंट्स वर हैविंग टू टाइप्स, outer integuments, outer integument and inner integument. Okay. Outer was forming testa, which is a part of seed, and tegma or tegment, which is a part of inner integument. Okay. They are the parts of seed. Okay. Fourth one, as you know, ovary forms. Before fertilization it was called as ovary, after fertilization it is called as fruit. And ovary wall, okay, ovary wall is called as pericarp. Okay, we will see it is a part of fruit only. Okay. And ovules is called as, we are seeing with what, ovules are called as seed, the mature seed. I hope you are clear with the things. Before fertilization what it was called as, and after fertilization what they are called as. Okay. Now we will see with the seed only, but seed are classified again in two types, okay, based on the layers, okay, albuminous and non-albuminous, okay, or it is called as endospermic and other is non-endospermic, okay, based on what they are, based on endosperm present in them as well as endosperm absent in them. Albumin is a type of protein, okay, which is present in seed. Now tell me, this is a which type of seed? You can see from this, when you are referring to this, just it is having dicot in it, so it is a dicot seed, how it looks, and for monocot it is somewhat like this. This is monocot seed. We have some layer in this. I have just drawn the rough diagram of it. In next lecture we will see with a deeper learning of this, okay, both the things we will see in detail, but just now, for now we just remember that seeds can be divided into two, that is albuminous and non-albuminous, okay, monocot and in dicot. Monocot example is maize, and for dicot mini are there, pea are there, okay, जो दो seed वाले होते हैं, and we can say with the again beans of those having with the two covering, mug and all the spices are mostly included in this dicot, okay. We will have a quick revisions of the today's lecture that we have seen with, okay. Today we have seen with what? Post fertilization event, that is endosperm formation we have seen, along with it we have seen with the example also, okay. Then we have seen with embryo we have seen with, and then we have started with the seed. Embryo formation we have seen in detail with the dicot, okay. In dicot how embryo formation is seen, I hope diagram would be there in this. Yeah, I will show you again the diagram. See, in biology diagrams are important. If you are clear with the diagram, then you can understand the topic more easily, okay. See, this is how the embryo formation takes place in dicot. Please try to draw the things that I am drawing on the board, which will be called as a running notes or rough note. Whatever you understand, जो भी आप देखते हो, try to see with the video 2 to 3 times, so that you can get with the things that were taught, and after that you can make your own notes by learning this, okay. See, this is how embryo were dividing, forming suspensory shell which was providing stalk, and embryo cells were dividing transversely and vertically, forming a proembryo, globular and heart shaped. I hope you are clear with this. Then we have seen with the events which were occurring before fertilization and the events which were occurring after fertilization, okay. Here you can see what were the things present before fertilization, that is pre-fertilization event, post-fertilization events were this, okay. Have seed coat, testa, tegma, fruit, pericarp and seed, okay. And for the next time we will see with the again structures, types of seed we will see with, okay. Thank you.